Last time the problem was I used my webcam. The unfortunate thing this morning is even though I came in last night and specifically inoculated a blood agar plate for you guys last night, I left it in my office before I walked over here. And I cannot teleport. I wished I could. Yeah, we don't have time for a field trip. So I'll bring them in on Friday, though. I figured out how to do the webcam, so I won't get all confused like I did last time. But so I'll actually have like actual media to show you guys as opposed to just the pictures in the PowerPoint. So as usual, I made a whole bunch of changes to the PowerPoint. I posted the newer version. And I'm going to post another PowerPoint for you guys that basically has like all the tables and figures in it from this new stuff. And so I only made one change to this PowerPoint, and that was to create a table to go over the information on virus-like particles. But I talked about this last time, so a little bit of a review, right? And you guys aren't telling me that you're not seeing it, but that's okay. So I will fix that. Who knows the answer to this when it comes up? If it comes up. I got coffee smoothie spilled all over me this morning. I'm still sticky. Guy walked right into me. All over the place. Mostly on him. I still don't understand why it's not showing up. The light is on, right? Projector's on. Light is on. I have it plugged in. Okay, so the third thing would be, is my computer projecting? And the answer is no. Yep, there we go. There we go. So who knows? The answer's there. Cover it up. Who knows the answer to this one? What's the fill in the blank on this one? So you have two choices in the picture. Neuraminidase and hemagglutinin. Which of them appears to be involved in the attachment to the host receptor site? Which one? H? Hemagglutinin? How many say hemagglutinin? Raise your hand. How many say neuraminidase? And it's hemagglutinin. Attachment, hemagglutinin, right? And actually, in our book, it uses the HIV virus as an example for attachment. Where's that attachment picture? No. It's hemagglutinin. Well, I'm glad if you wrote it down wrong. Look, right here. HA, hemagglutinin. HIV virus, that's what it uses to attach. The neuraminidase is what shuts down the receptor that hemagglutinin binds to once the virus gets in. And that's how they make it so that only one virus gets into one cell. But it is the hemagglutinin that actually binds to the receptors on our cells. And also remember, like, heme refers to blood, gluten refers to clumping. This will actually cause your blood to clump, too. Which makes sense because it's binding. It'll bind to red blood cells. Okay, so now I want to move on to, and so this table I'll post it in another file for you guys with all the tables and figures that are new. So, viroids, what type of nucleic acid are they? Who found out? They're RNA. And in fact, they are just RNA. They do not contain any protein, none. They're just single-stranded RNAs. But they will bind to themselves. But this isn't double-stranded, right? It's still single-stranded. And so we have a picture of that coming up. Why have you probably never heard about these before? What host do they infect? What cells do they infect? Plants, right? Which is probably why you've never heard of them before. unless you're a botanist, right? You're supposed to stick to human stuff. So an example, right, and again, I'm not going to make you memorize examples, but sometimes examples help us. The example they talk about is the potato spindle tubular viroid, and it causes deformity in the potato. So formerly known as viceroids, now divided into two groups, satellite RNAs and satellite viruses. So we're going to keep this in general terms because this is even relatively new to me, and I've been trying to do some research on it. I haven't had time to really get all the answers to all the questions that even I have, right? So certainly don't expect you guys to have a firm understanding of these guys. But in general, right, what type of nucleic acid? Well, the name tells you, right? Satellite RNAs are RNA. But they also contain protein. So very much like a virus. So why don't we just call them viruses, right? They're nucleic acid, they're RNA, and they have a protein coat. Wouldn't that make them a virus? The only problem is that they can't infect cells on their own. They need a helper virus. Their RNA doesn't actually encode for their capsid proteins, for the protein, that protein coat. That RNA doesn't actually code for that. It's a virus, what we call a helper virus, because it helps out these viceroids, actually encodes for their protein. So again, probably never heard of these before because they infect plants. And I don't even have an example for you. They don't give an example in the book. And the ones that I found have weird names that I don't think would help you remember. Okay. So the next one, satellite viruses. So again, why isn't it a virus? Right? Why don't we just call it a virus? Well, just like satellite RNAs, viceroids, right, what we commonly refer to collectively as viceroids, are RNA. The difference here is this guy, its RNA, actually codes for its own protein code. So, okay, well, again, why isn't it just called a virus? Right? A virus is nucleic acid in a protein code. At the very least, a naked virus is that, right? So, why isn't it a virus? Why is it a satellite? Well, it's called a satellite because guess what? It too cannot infect cells on its own. So the example kind of gives you a clue as to where you might find these. The hepatitis D virus, and some people call it a virus, or some people call it a satellite virus now, or some people used to call it a viceroid, right? We're still trying to tease this stuff out. So who gets hepatitis? That would be us, right? Animals, right? But plants can get these satellite viruses as well. My understanding of this was that basically you have the hepatitis B virus, but inside of it, you have this RNA with its own protein coat that we refer to as the hepatitis D satellite virus. So basically a virus has been infected by a virus. In this case, a satellite virus. Why do we call it a satellite? Again, it can't infect cells on its own. It needs the envelope in those proteins that are made by the hepatitis B virus. So you can't get hepatitis D without B. It's a co-infection. So when scientists first discovered hepatitis D, they thought it was a brand new virus. So again, they named it hepatitis D. It was the next letter in the alphabet of the hepatitis viruses, right, that we deal with. But further investigation they found was like, well, it's basically B, but it has this other piece of DNA, other piece of RNA, excuse me, in there that encodes its own protein. But it itself can't do it on its own. So it's like, you know, it's one of those blurred lines. Do you guys see why, like, even I have trouble grasping this? So at this point, I don't expect you guys to either, right? But you know, as we can go down to this molecular level, right, where we're actually looking at code of viruses, right, and what's coding for what, and where the proteins are coming from, and how these things interact. As our advances in studies increase, we're finding things that we probably never imagined was possible. So this is relatively new area, as I said. Even for me, I don't have a good foundation on it just yet. The important thing with viceroids is remember that these are RNAs, basically, that are found in other viruses, in what we call helper viruses. The only difference between the satellites versus the satellite viruses is that the satellite viruses actually encode their own protein code. Unlike the satellite RNAs, rely on that helper virus to actually even make their protein code. The last is prions or prions. It's pronounced both ways. I very easily usually just say infectious proteins, because that's actually where that name prion or prion comes from. So, do they contain any nucleic acid? No, they're just proteins. They're just protein. But as we know, proteins can be catalytic, right? They can catalyze reactions. So we found these in animals, and of course humans are animals. And in general, these are a group of proteins that cause what are collectively referred to as transmissionable spongiform encephalopathies. And so encephalopathy refers to the brain, right? Spongiform, like, it literally makes the brain look like it has holes in it. It causes destruction to areas in the brain. So progressively, eventually, causes death in the animal. So some other common names you may have heard for this. Anyone know a common name for this condition that happens in cows? Mad cow disease. It's called Christoph. It has some two-person name in humans. It's called scrapie in sheep, but it's all the same thing. And again, our understanding is not very deep as it relates to these. We know it's a protein that is causing proteins like itself to deform. And because those are abnormal deformed proteins, they no longer do their job. The really bad news is these proteins are in your brain, right? And so having your brain lose function progressively over time leads to death of the individual. And of course, if the brain's affected, the cow acts strange, right? Hence the name mad cow disease. The bad news is in England one time, some brainiac thought it would be just brilliant to feed cows to cows. So literally the cow died, they turned it into food for other cows. Okay, cows don't eat cows. Even when they die, right, they walk around them, they eat the grass next to them. They don't eat cows. They're not omnivores. They don't eat meat, right? Somebody's like, oh, just recycle. Not a good type of recycling, right? So unfortunately, some of those cows that died had this disease, right? Had these mutated proteins, right? They believe for people too that a mutation could happen, and that's where this probably came from. There was a mutation in these proteins. The bad news is that if you ingest these proteins, they cause your proteins to then mutate, right? Or deform, you should say. not mutate, but deform. Mutation refers to a change in DNA, right? Does that make sense to you guys? So again, they're still trying to work out, all the details in this. So for viroids, right, we're talking about RNA, and it can bind with itself and form these really cool structures. But it is single-stranded, and it's just RNA. And we just really see these in plants. And again, this is an active, relatively new area of study. And then the hepatitis D virus, that satellite virus that's found inside of hepatitis B. And then other satellite RNAs that don't even encode their own protein coat, rely on a helper virus. These are found in both plants and animals, the satellite viruses. And the satellite RNA is mostly just plants. So here's poor, sad-looking cow, right? And so, again, no nucleic acid, no genes. It's just a protein, right? They believe where it came from was literally a mutation happened in this protein in the DNA. And unfortunately, the new form of this protein causes normal forms of the protein to become abnormal. Right? So it's like a catalytic reaction. The bad news is these proteins are in the brain. Right? So it progressively deteriorates, destroys the brain, and kills the animal. And so it's got, the disease itself has lots of different names in different animals, right? But collectively they're known as transmittable spongiform encephalopathies. So that's it on our viruses, which brings me back over to our growth and control. So let's see how you guys did on your homework. So you need to be able to list the 10 elements that we require in large amounts. And specifically, we need to know which ones are found in proteins, carbohydrates, nucleic acids, and lipids. So I had posted that table, right? I have the filled-in table for us today, right? So let's first start out, what are those 6 major ones that you find in proteins and carbohydrates and nucleic acids and lipids? So these are elements on the periodic table, right? So C for carbon. That's the main one, right? And we talk about where organisms even get that from. What would be another one? Hydrogen, oxygen, phosphorus, sulfur, one more, nitrogen, right? Okay, and then there are 6 elements that we need in small quantities. Anyone know why we don't need a lot of them? Well, no. Well, they could kill us in large quantities, you're right about that, as well as microorganisms. They're not part of the building blocks for making these molecules, right? Those are the ones we need in the largest quantities to actually be able to make these molecules, right? Or organisms need to make these molecules. So they're things that aren't as abundant. So we don't need as much of it. But we do need them for cellular function. So you guys name the big 6, right? Top 6, carbon, nitrogen, phosphorus, sulfur, and oxygen. Where did hydrogen go? Should be, that's only 5, right? Ah, where did hydrogen go? What did I do? I don't know, but add it in. It wasn't, yeah. Shame on them, or me for not noticing that. Okay. That one's up there. Okay, other macro nutrients that would round out the top 10, and it varies from, source to source, but things like potassium, and this is the one that throws most people off because the abbreviation is a K, right? And then sodium, magnesium, calcium, and iron in the different forms. And notice most of these, how they're written, They're written as what we refer to as cations, where they're positively charged. They have a tendency to very readily lose their electrons, and so they're not balanced. Unlike chlorine, you usually see it in its ionic form, and it has a negative charge. It's what's referred to as an anion. It readily accepts electrons from other elements. Right? It's a little bit of an electron hog, you could say. But these are important, right? If you've taken anatomy and physiology and stuff like that, these are really important, right, into the functionality of cells for ionic balance. Things like muscle contraction, right? Moving the actin filaments around, all that fun stuff. So same thing for microorganisms, right? Ions and ionic balance are really important to the functionality of chemical reactions within cells. So these are needed in relatively large quantities as well, but not quite as much as the top 6, because the top 6 are actually the building elements that make up the molecules that are the structural and functioning parts of a cell, right? Proteins, lipids, nucleic acids, sugars. Ones that you need in smaller quantities, and most of the time it's because they're needed for additional structural support in proteins, and a lot of times in the activity of them. So for instance, when we copy DNA in a test tube using special enzymes, right, in a reaction called polymerase chain reaction, PCR, we have to actually make sure that we add magnesium to that solution, because the enzyme will not work without it. It's sometimes what we refer to as a cofactor, right? It's needed for that reaction to go forward. So sodium again is listed again down here, but magnesium listed again, manganese, copper, zinc, these type of metals are necessary as cofactors a lot of times for enzymatic reactions, where a protein needs this element in addition to actually do its job, right, to catalyze a reaction, or even hold its specific structure it needs to have to do its job. There's one on this list that probably looks very foreign to you. Right, I have it highlighted in green. Molybdenum. It's a mouthful. Abbreviated MO. How many of you guys have ever even heard of that element before? How many people take a multivitamin? Do you actually know what's in the multivitamin? Have you looked at the list of elements or vitamins? You might want to do that. Nowadays, they've been adding this to supplements for people even. They're not exactly sure what it does or what it's needed for, but apparently it is beneficial for even people. Most of the time we have to provide this for certain microorganisms that do a particular process known as nitrogen fixation. This is converting nitrogen from nitrogen gas into ammonia, which is very important in our ecosystem, is this cycling of nitrogen through the system. So when we look at these, right, when we look at sugars, when we look at amino acids, when we look at nucleic acids, you will always find carbon at the backbone, at the center, you could say, right, that everything attaches to. So the answer is yes for all of these for carbon, right? But let's go down the list. Do carbohydrates, right, which are sugars, and by the way, carbohydrate means hydrated carbon. Hydrate is water, right? What is the chemical formula for water? Everyone knows this one. H2O. So guess what the answer is for this one? Carbon, right? Oxygen and hydrogen. Carbon and water. That's an easy one. Does it contain any nitrogen, phosphorus, or sulfur? Carbohydrates, sugars, no. Amino acids, do they contain carbon? Better say yes every time when I say carbon, right? Do they contain oxygen? Yep. Do they contain hydrogen? Yep. Do they contain nitrogen? Yep. Do they contain phosphorus? No. Do they contain sulfur? Yes. Most of them do, and right, this is why she's questioning it, because most proteins contain at least one, if not more, of 2 amino acids that contain sulfur. Only 2 of your amino acids contain sulfur. They are cysteine and methionine. And if you remember back from genetics, especially for bacteria, their very first amino acid they put in a protein is methionine, right? Because that's the start codon, is methionine. So for nucleic acids, which are their individual building blocks, right, or nucleotides, do they contain carbon? Yep. Oxygen? Yep. Hydrogen? Nitrogen? Nucleic acids, nitrogenous bases, right? You hear all these N words? Yeah. Phosphorus. Yeah, right? It's the sugar phosphate backbone that hooks the nucleotides of DNA together, so that's a yes. Does it contain sulfur? No. This is the only no for nucleic acids, is sulfur. So if you wanted to tell proteins from nucleic acids, you could look for phosphorus versus sulfur, right? And figure out whether you're dealing with a protein or a nucleic acid. Because nucleic acids contain phosphorus where proteins do not, and proteins contain sulfur where nucleic acids do not. Fatty acids, right, that make up things like lipids. So things that make up our membranes. So do they contain carbon? You betcha. Oxygen. Yep. Hydrogen? Yep. Nitrogen? No. No nitrogen. Do they contain phosphorus? Yeah. Some of them do, right? Because what are our membranes made out of? Phospholipids. So yes for some. Oh wow. It's like, makes sense, right? That's why we call it that, right? It's what it's made out of, right? Literally at the element level, right? We're being specific about that phosphorus. And again, it's because it makes that area positively charged, right? It's kind of a greedy or not so greedy electron. And then it doesn't contain sulfur. What? Oh. You were ready. You did your homework. All right. So this table, as it is like this, I also put in a separate PowerPoint that I'm going to post for you guys. So you don't have to click through. You can just have like it and print it out if you want with blank ones. Okay. So Lance already jumped ahead and did his homework, which was good, on this unit, which goes over these definitions. So we started to go over them last time, and I find, too, it's like you got to practice this stuff, right? So I had asked for you guys to try and fill in the table, right? So when we talk about organisms, we talk about where they get their energy from, we talk about where they get their electrons from, and we talk about where they get their carbon from. And for each one of these, we have terminology. And for each one, we have 2 choices. So for energy source, there's 2 choices. And we put these all together into one nutritional type. And remember I said, right, so each one of these, energy source, you have 2 choices. Electron source, you have 2 choices. Carbon source, you have 2 choices. So I should actually have a greater number of combinations here, right? 3 times 3 times 3 would be what? Can't think of how many different combinations. But why do I only have like 5 written here? Some of these tend to go hand in hand. right? And that's where Lance was getting confused because they were saying, you know, some organisms that are heterotrophic are also probably organotrophic, right? So they have a tendency, right, to have these similarities and to use molecules for multiple means. So I'm going to jump to the bottom of the table for a second. This would be us, chemo-organo-heterotrophs. We use sugar for all of these. We use it as energy, we use it as a source of electrons, and we use it as a carbon source. So that's where the confusion is. We'll use the same molecule to do everything. Get energy, get electrons, get carbon. But when we're specifically saying, right, an organism is a phototroph, we're referring to what? Energy source, right? And that specific energy source is light. Then when we look at the next, right, when we say litho, we're referring to electron source, right? And in that case, what does litho mean? Inorganic source of electrons. There are rock eaters, right? So imagine something not living. Their carbon source, if they're an autotroph, right, which is the next section of their name, they're getting their carbon from what? No, not the sun, but something in the atmosphere. Carbon dioxide. So yeah, these are plants, right? Plants are chemo, I'm all right. Photolithoautotrophs. So are photosynthetic cyanobacteria, other photosynthetic bacteria, algae, and these are the ones through this process actually produce the excess oxygen that make life on Earth possible for us, right? Because we need that oxygen. The next one is photoorganoheterotrophs. So photo refers to, again, where's their energy coming from? It is light. Organo tells you their electron source is what? Organic. That one's in its name. Isn't that nice? Right? And then heterotroph, where are they getting their carbon? From an organic source. So as you can see here too, do you think where they're getting their carbon and electrons is probably the same thing? Yep. All right. So it makes it easy if you're getting two of the things you need from the same molecule. So chemolithoautotrophs, in this case, their energy is in a chemical form, right? They're not using light. They're using some type of molecule to get energy from it. You don't know what type that is until you look and see where their electrons are coming from. Because if they're using chemical energy, right, that molecule they're using for the energy is also where the electrons are coming from. So you gotta, if they tell you where the electrons are coming from, you actually know what type of chemical energy it is specifically, whether it's organic or inorganic. So if this is a chemolithoautotroph, its electrons are coming from what type of source? Inorganic. So their chemical energy is inorganic. And then where's the carbon coming from? Carbon dioxide, right? They're autotrophs. It's carbon dioxide. So you see, Lance, how some of these are sometimes linked? Yeah. But you do have to look at each one separately. So chemolithoheterotrophs, so again, it's inorganic, right? Because they told us it's a lithotroph. It's an inorganic source of electrons, so therefore the energy is also an inorganic source of some molecule, some chemical. But these guys are heterotrophs, so they're getting their carbon from what? Organic source. And then the last one is us, right? It's all organic. And so the example is, I mean, we use sugar for all of this. So this is isn't in the PowerPoint that I posted this morning, because when I was flipping through it, I realized I didn't do what I told myself to do, was insert this from chapter 13. So they referred to this. And again, I always like to give you guys like examples, but by no means do I expect you to memorize them. But for some of us, it helps us really kind of, be like, oh, that's what that is. So we are chemo-organo-heterotrophs, right? But so are most bacteria, so are most microbes that aren't photosynthetic. Basically, anything that's not photosynthesizing is probably a chemo-organo-heterotroph. It's getting everything from some organic source. Where you get into the really kind of cool, I think, microorganisms is when you get into ones like these guys that are using chemical forms of energy that's inorganic, but they switch up where they get their carbon from. And so notice, I said we're stuck in our category, right? And plants are stuck in their category. That's pretty much all they can do. But some microorganisms can switch depending on what's available. Like they have the ability to do more than one thing. So sulfur oxidizing bacteria will utilize sulfur, an inorganic source of electrons, when it's available. They're called sulfur because they use an inorganic source of electrons, but their carbon can come from organic sources or from carbon dioxide. So they can switch between those two. That's pretty impressive. Why would you want to be able to switch? Well, think about a photolithoautotroph, right? A plant. What must it have to exist on this planet? It's got to have sunlight. If we take away sunlight, what's going to happen to that plant? It's going to die. In the reverse, right, for us, can we photosynthesize? No, right? Otherwise, we'd probably look like green Martians, right? We'd have chloroplast just like plants do. And we could tell our parents, I don't have to eat my vegetables. I'm just going to make my own sugar, thank you very much. I'm going to go sit outside in the sunlight. Not going to happen, right? We can't switch like that. We're stuck. And because we're stuck, right, we've got to eat our vegetables, right? We've got to get our food in a particular manner so that we can utilize it. Right? We can't take carbon dioxide and light energy and make our own sugar. But some microorganisms can kind of switch between classifications, which is great for them, right? Because if the environment changes, they're like, okay, I don't have anybody to eat, no organic source of carbon right now, so I'll just switch to carbon dioxide, no problem. That's readily available in the atmosphere. I'm set. Right? So a lot of them are stuck like us, but there are others, like especially the sulfur oxidizing, they can switch between their carbon sources. Right? And there are some organisms that can switch between photosynthesis and using chemicals as a source of energy. So again, if the light goes away, they're like, no problem. I'm just going to use the sugar I stored over here for a dark day. Make sense? Right. But do we need to know this about the organisms that we're working with? Like what they need and what they don't need? Yeah, especially if we want to grow them, right? If you took a photosynthetic organism, you put it on a petri dish, and you put it in the incubator, and you close the door, when you come back tomorrow, is it going to grow? Got news for you, my incubator has no light inside. It's not going to grow. It needs that light energy, right? That's why one of the organisms we don't work with, because I don't have a light in my incubator. Right, so remember that each one of these, when we talk about phototrophs or chemotrophs, we're talking about energy and where that energy is coming from. Is it light? Is it chemical? And then if it's chemical, if we know where the electrons come from, we know if it's inorganic or organic, right? But if they just say chemotroph, you just know it's a chemical form. You don't know if it's organic or inorganic. So the electrons are a little bit linked, especially for our chemotrophs. If they tell you where the electrons are coming from, organo, that's easy, organic, right? Litho is inorganic. Think rock eaters, right? Think not alive. When they say Autotrophs or heterotrophs, we're talking about where is the carbon coming from. Is it organic? They're heterotrophs. Or are they autotrophs? It's carbon dioxide. So as I said, nitrogen, as we saw in our table, is really important, right? It's part of amino acids. It's part of nucleic acids. So we need it for our cells. So cycling that element through the ecosystem is really important. And microbes make that also possible, right? If it wasn't for microorganisms in the soil that convert nitrogen gas into ammonia and usable forms that plants can use, that animals then can eat, life on Earth would not exist. We wouldn't be able to get nitrogen without that. So we need those nitrogen fixers. And they need molybdenum. I need that element to do this process, or they can't do it. So we talked about autotrophs already, right? So these are oxytrophs, oxotrophs. These are ones that they may have had a mutation, and that if you don't give them a particular food, they'll die because they can't synthesize it themselves. So a lot of times it's like they're defective in being able to make certain amino acids, right? They can't put that together on their own. Where prototrophs are great because you can give them inorganic everything and they'll make everything they need. You don't have to give them anything preformed. You don't have to give them amino acids, which are organic molecules. You can give them nitrogen source and a carbon source, and they'll turn it into whatever they need. So the next set that we're going to look at, and I'm going to go through really quickly, but hopefully you guys are familiar with it. And as I said, if you haven't filled out these tables already, try before you even look at my answers. Try to fill in these tables. Try to think about each one of these categories in these terms, right? Does it require, does it have to have oxygen? Can it grow in the presence of oxygen? Why is that a question, though? Why do we ask that? Can it grow in the presence of oxygen? Oxygen's awesome, right? For us, right? We need it and we can protect ourselves from it, right? Oxygen can form toxic forms. And especially when you're utilizing oxygen for your metabolism like we do, we form toxic forms of this molecule all the time. We form hydrogen peroxide and superoxide radicals. Why don't we die? These are toxic. We even make them, and they're toxic. Why aren't we dead? The answer is one of our questions. We have enzymes that convert the toxic forms into safe forms of oxygen. So the question here is, can they grow in the presence of oxygen? Should be more like, do they have protection against oxygen? Right? Are they going to survive that exposure? And then, of course, can they grow without it? Right? So some organisms, do they have to have it for their metabolism? No. Some of them can grow without it. They have other means of acquiring energy other than using electrons donated to oxygen. So, let me see. What else did I want to say? So these tubes, I'm even going to have examples. I'm going to actually bring the real thing from lab on Friday. So, I was kind of hoping to do show and tell more today than anything else. Make sure you read through. I did make changes to this, and I will post stuff. The one last thing that I want to say is the very last objective on your list actually isn't in Chapter 6. So for Friday, I want you to read, and I posted it. Yep. Yeah, the last objective is in 2 different chapters. So look under modules where the PowerPoints are listed. It'll tell you what sections in your book to read for Friday. It's right here. I'm going to click on it so everybody can see it.
Did you remove some of the things from the PowerPoint or just add new things?
I moved stuff around too, right? So the last one is chapter 15, section 1, and chapter 11, section 5. So it'll be like 4 or 5 slides that I'm going to add.
I'm also in the lab.
Mm-hmm.
It's like right now, I'm going, but I can't even stay awake. Is there, like, my head just,
If you guys want to check in late, is that what you're standing up here for? What's your name? Group. Say again? Perkins.
Is there a makeup day for this lab?
No. I got you. Andrea helped me, so thank your friend. Okay, I just realized I marked her as absent instead of late. So Stephanie Stewart. Did she run off? Yeah. She's not going to be able to understand what I would tell her right now if she can't stay awake. Huh? She said she's not coming to lab. Inside out.
Yeah.
Alright, the only thing that I did put on your guys' lab benches that you need today is a striker. So if someone from each group wants to come pick up a striker, that's actually not the only thing, but it's one of the things. Alright, so there's 3 separate handouts that you're going to need for lab today, so go ahead and pick those up. Oh, bless you. Oh, did not type a password.